Hi, everybody. So, Matt Disney. I'm a professor at University of Florida Health. Uh, it's a biomedical research institute. So I live next to the beach, and so I was thinking when I was about this talk about what morning picture I can take. And so I think we're on the precipice of a dawn of a new day in the treatment of ALS, and I know things take long. Um, and too long at times. But I think that we're nearing an era where the dreams of getting medicines are going to hopefully turn into a reality to help people. So I'm going to sort of give you a lay of the land here about C9orf ALS, so chromosome 9 open reading frame 72 ALS. That's also associated frontotemporal dementia. So I'm going to speak in a very basic way about medicine development in this landscape of diseases, and then have some views of my own work. But I want to sort of level set you guys for what I do. So I am a professor. I run an academic lab. So that's my job. My job is to teach students that are in the lab. I have anywhere between 20 and 30 graduate students and developing scientists in the lab that work on these research projects. And the way that we operate is we get funding from the National Institutes of Health to be able to do this work. And my output is the science that I'm going to tell you about today, but also the people that are in this slide set. And so we would not be able to do any of this if it wasn't for the US taxpayer dollars, especially NIH funding that you guys pay taxes, and that tax dollars gets allocated for people like me to do research. And as you'll see later on in this talk, that research can hopefully inspire other people in the lab, other people outside of the lab to develop medicines for diseases. And what's common about my lab, but might be uncommon about you guys in doing science, is that science is an international affair. And so in my lab, I have postdocs like Amir, who's from Iran. I have postdocs like Patrick, who's from Germany. I have visiting scientists from Japanese pharmaceutical companies like Wataru, and scientists like Raphael from Israel. And they all come to learn science on what we're doing and to take what they learn to a drug company, or like Raphael is doing at his own lab in Israel. Okay, and so C9 ALS is an autosomal dominant disease. And what does that mean? That means that if we look at our genes and chromosomes, which are shown here, is that if you, for example, have an affected father that has one copy of a mutated gene, if they have, your father and your mother have 4 children, then the chances are that 50% of them are going to have that affected gene. I'm one of 7 children, and so you can imagine how these genes are passed on. That means if my father has a gene and it's autosomal dominant, which means you only need one copy of that gene to exert an effect, well then, 3 or 4 of my siblings would have that gene. And so I'm going to tell you about C9orf ALS. And so I don't know the level of the people that are on this call, but I'm going to try to make it very basic. And so this is a large RNA repeat expansion, which means a repeat expansion means it's just you have a segment of your DNA that has a certain sequence, and that sequence expands to be very long. Well, this repeat expansion that causes C9 ALS FTD is present in chromosome 9, open reading frame 72. So that means it's on the 9th chromosome, and it's in the 72nd gene on that 9th chromosome that could be translated into a protein. Well, that gene was discovered in 2011, and we're in 2023 now. And so that's a long 12 years, I realize, but you should think that the basic scientists and biomedical researchers that helped discover that gene in 2011, I don't think that they would have dreamed that there would be compounds in phase 2 clinical trials against this gene product just 12 years later. It's still not fast enough, but I do think that it's hope and that things are developing. And this and what's good about this is there were two independent papers. One where the last author, the corresponding author, the scientist who lab was directing the research, was Rhoda Rodemakers, who at that point was at Mayo Jacksonville. And the second principal investigator was Brian Trainer at the National Institutes of Health. And so how were they able to find this gene? ALS is a disease, and C9orf72, ALS and FTD, was not just came to be in 2011. It's that it gets very difficult to find genes that cause diseases. It's much like finding a needle in the haystack. And so Brian Trainer and his group were able to find this gene because they did genome sequencing and analysis of a population of Finnish ALS patients that had both ALS and frontotemporal dementia. And by sequencing Finnish populations where you can imagine they're not as genetically diverse as you or I, if we're Americans, it was much easier to find. If you don't have a lot of genetic diversity and one person has ALS and FTD, it's easier to figure out what gene is causing that through a population where it doesn't have, as much diversity as you might have in the United States. This was found to be the most common mutation known to cause familial FTD and ALS. Disease is caused by a large repeat expansion, so it's greater than 100 or so repeats. The repeats are between 50 to about 1,000, and healthy repeats are 20. So it's literally, like I said earlier, just a large repeat expansion. And subsequent studies, including those by Jeff Rothstein, Len Petroselli, Paul Taylor, and a wide variety of other scientists, was able to identify multiple mechanisms that cause disease. And so once you know a gene and you know the mechanisms by which that disease is caused by that gene, you can design ways to intervene with therapeutics. And so what was exciting for this is that this is an RNA repeat expansion, which means that in a messenger RNA, you have a large RNA repeat. There's some historical context for RNA repeat expansions causing diseases. And so when I was preparing for this talk this morning, I went and got this paper. This is a review article by Maury Swanson, who's at the University of Florida. And this is just a list. Each one of these lines here is a different disease that's caused by a different RNA repeat expansion. And so there are some important diseases that are here that you might have heard of, including Huntington's disease. Another one is myotonic dystrophy, which is the most common adult onset form of muscular dystrophy. One is Fragile X syndrome, which is the only known single gene cause of autism. And so I think there's a major advantage in knowing that C9orf ALS is caused by an RNA repeat expansion. And that's because there's greater than 50 diseases that are caused by these repeat expansions that have varied mechanisms. And this affects, in total, each one of these individual diseases would be considered a rare disease. But I would, posit, and I'm sure you would agree, is that these diseases are not that rare. These affect millions of people worldwide. In fact, for these 50 diseases, if we just assume an incidence of one person in 10,000, which is probably conservative.
I was curious as to how many of those diseases on the list there actually have any significant treatments or cures, because, even though that it's a repeat expansion, just because it's a repeat expansion doesn't mean that it all does the same thing. But, you have to make the proteins, and the proteins can all do whatever they feel like doing in each of the disease processes. So I was just wondering if that whole list. Yeah,
none.
That's what I thought.
But this is what I want to say. So DM1 has probably 20 compounds that are being put into clinical trials, and at least 2 that are towards the end of a phase 2 clinical trial. Huntington's disease has multiple compounds at multiple different approaches that are being put into clinic. These include oligonucleotide medicines and small molecules that can eliminate the toxic RNA repeat. And there's Friedreich's ataxia has a wide variety of compounds, and so that are put into clinic. And so even though right now, that's one of the themes for this, even though right now these things aren't approved, when clinical data is collected for each one of these indications, it's going to be easier to get a disease for C9 ALS because it's going to be information that can allow you to better target that disease. Does that make sense? Can you guys hear me? Okay.
Yes,
No?
I can hear.
Okay, good, because I'm trying to make sure I don't, because I'm sharing the whole screen. I can't tell if things crash. Okay, so, all right, these are 50 diseases with varied mechanisms. It affects 40 million people worldwide based on an incidence of 1 in 10,000. And so as we are aware, DNA gets made into RNA, gets made into protein. And so in C9orf ALS, you know, that is chromosome 9 open reading frame 72. And so that means the DNA is in chromosome 9, it's open reading frame 72, which means that gene on the DNA is in an open reading frame that ultimately becomes a protein. And so that DNA repeat gets made into an RNA, that RNA gets processed, and it gets translated by the ribosome. And so there's a wide variety, because we know that information for how this DNA repeat is processed into RNA and ultimately protein, biologists were able to triangulate a few mechanisms by which the disease could be caused. One is a loss of function. That means that the C9orf72 protein, which has an unknown function, loses that function. And so it will have an important function in a nerve cell, but we just don't know what it is. But the other mechanisms are the ones that are being most pursued for therapeutic intervention, and they're what's called gain of function mechanisms. And so in those mechanisms, the RNA is made. And in an RNA toxicity mechanism, the RNA becomes toxic in that it binds to and sequesters proteins that normally affect other genes. And so when the repeat expansion binds them, those proteins cannot go on to affect the processing of other genes. And so you get major defects in RNA in neurons. The other thing that can happen is those, as I suggested earlier on, is that these RNA repeat expansions can be translated into proteins. And those proteins themselves are toxic and could cause neurons to die. So most of the C9 ALS targeted medicines have focused on affecting RNA-associated defects. And so these defects include, as I mentioned earlier, translation of these toxic repeats into these toxic repeat RNAs into toxic proteins, affecting the ability of these large RNA repeat expansions to sequester proteins that are involved in the processing of other RNAs. But also, it was found by a variety of researchers that in ALS-affected genes, ALS-affected cells, in a variety of systems, that you get defects in what's called a nuclear pore. And so basically, your nucleus, where your DNA is stored, has to translocate RNAs that are made into the cytoplasm, so they're made into protein. Well, some of those proteins that get made in a cytoplasm have to come back in the nucleus where their function is. And what regulates the entry and exit of those proteins is what's called the nuclear pore. And so it was discovered by a wide variety of researchers, including Paul Taylor, Jeff Rothstein, and others, that a unifying principle in many of these repeat expansion disorders is that you have a defect in the nuclear pore. And so although I'm not going to talk about it today, there was a drug put in clinical trials for ALS that would affect the nuclear pore that ultimately didn't reach a clinical endpoint. And so what I'm trying to tell you is that knowledge about the gene, the biology of the gene, can inform ways to directly target the toxic gene, but also ways to develop a medicine to affect the downstream processes that could be causing a disease. And so so the goal of this talk for the end of this is to describe work on how to disable RNAs. And so what I'm showing here is in purple or violet, whatever this color is, an RNA structure. And so there's two sort of regions of an RNA. One is if it's shaped like this, it's folded. So it's folded like you would wrap a present. It has a very defined way that it's packed. Or if it's like this, it has a sequence that's just flapping in the breeze and doesn't really have a defined structure. Sorry. And so based on these folds, there are different ways to bind to and disable the RNA. One is what my group does, and the others is what some other people have done in the clinic. And so my group, we're an RNA group where we try to, see, this is a case where I can't spell RNA. It's RNS, but it should be RNA. You figured RNA would be easy to spell. And so what we do is we design organic molecules called small molecules. These are things like aspirin that you can take as a pill, and they get to a disease-affected tissue and even crossing the blood-brain barrier, is that these molecules bind to these RNA structures that cause the disease and disable them. The advantage of, they have multiple ways to affect the RNA's biology, and they can ultimately be delivered as oral medicines. The disadvantages of these is we're very much at the beginning of using this as a strategy, and so there's many unknowns. And even if we know the three-dimensional structure of this RNA, structure-based design can be challenging. Now, the other way to target an RNA sequence is this antisense or siRNA-based approach, where basically you can very easily design pieces of DNA that recognize these unstructured regions of an RNA and recognize them by using Watson-Crick base pairing. So you know that A binds with T and G binds with C, and you can custom synthesize, even on a machine, molecules that are recognized to a predefined sequence in this RNA target. And that binding, as I'll show you later, of this oligonucleotide, antisense oligonucleotide siRNA to the sequence will bind to and disable this entire transcript. Now, the disadvantages of this approach is that it can have dose-limiting toxicity. Now, when the molecules are injected, these molecules have to be directly injected into your central nervous system. And so if they have peripheral dose-limiting toxicity, that might be not much of an issue if you inject it directly into the CNS. And so, what I'm going to describe are efforts that have been used to ASOs that have been put into the clinic. And then towards the end of this, how we've been designing ligands or small molecule lead medicines that target RNA structures and what the implications of those are. So I sort of showed you how C9orf ALS gene was described 12 years since it was identified in patient populations. They understood generally the biology for how the disease is caused. And because you have a gene, you know the biology that the gene is causing, you have what's called the potential to develop a targeted therapeutic, which means you can develop a compound like a small molecule that we described on the previous slide, or a piece of synthetic DNA that can bind to a gene and disable it. And so here's one of the things that I want to press you. So this morning I got from this is that this is from Clinical Trials Arena, is that the number of phase 1 to phase 3 drug trials in ALS is expanding dramatically. So even from the time when C9orf was discovered to now, the number of clinical trials in ALS is 3 times increased. There's another thing I want to show you here. When you see this maroon color in company, that can often mean that it's a biotech company or a large pharma. And the amount of change in this number of trials is mainly driven by pharma getting involved. Why would pharmaceutical companies get involved in this? They are betting that they can get a medicine for this. And so the sheer fact that there's a large number of clinical trials means they know more information about what the targets are. And they also have the ability, which I'll talk about later, to know if a molecule in a patient, a human, will modulate the target, what's called a biomarker. And I think, these numbers are going to continue to increase. I think, and it's ultimately going to lead to multiple compounds for multiple different types of ALS as being FDA approved. Now, I ended my introduction for ways to disable RNAs by talking about oligonucleotides. So oligonucleotides have been put into humans as a treatment for C9 ALS by eliminating the toxic RNA. And so more detail about how these compounds work is that they bind to a toxic RNA, and then they recruit endogenous ribonuclease H, which is basically a scissor. And basically, it by forming that oligonucleotide, basically piece of synthetic DNA that binds to the region of C9orf message, you can engineer cells to cut the C9orf message in positions and eliminate it. And so 2 clinical trials for oligonucleotide-based therapeutic for this form of ALS were put into trials. One was a phase 1 study by Biogen Ionis, and one is a phase 1b, phase 2a study. And the results of these studies in the form of a press release are shown here. Now, these are top-line results, which is basically a description of the summary of the results without too much detail for what's going in. They were disseminated maybe 6 months ago, maybe a year ago. And ultimately, Biogen decided they didn't want to continue this program, but Wave did. And what a phase 1b/2a means is that you're studying in patients whether the compound is safe, but also if it's efficacious. And so what Wave did is they designed oligonucleotides that can basically decrease this gain of function mechanism that I showed earlier. And so this is published work, and I took this directly from Wave's website. And so you can go under Wave Therapeutics website, and they have corporate documents, and this is slide 34 of their corporate document. And so what they can do is they can deliver these synthetic pieces of DNA, and then they can use poly-GP as a biomarker to basically, poly-GP is a protein that's made from these repeat expansions, and they can basically determine if delivering of these medicines reduces that biomarker. And that would be a proxy for the compound they designed is hitting the target and eliminating it, and it's diminishing the production of that toxic protein. And so here's some of the results in a in vivo animal after 6 months of dosing. And so what they were able to achieve in a spinal cord and a cortex of a mouse is that you could get greater than 90% reduction of this poly-GP dipeptide repeat. And so dipeptide repeat is just the toxic protein over time. And that happened in a manner where you didn't have any effect on C9orf protein itself. You were just diminishing the amount of the toxic part of the peptide that was produced from that intron in that RNA. Okay, and so this molecule was taken from mouse to human, and what they did is a single dose study in this phase 1b/2a study, and so they could show that in humans they were reducing. So they tested 2 doses: a 10 mg per kg dose, a 10 milligram dose, and a 30 milligram dose in man, and you could see that this poly-GP biomarker in cerebral spinal fluid from patients was reduced through day 85. And so that compound and that data is sufficient for Wave to continue their clinical trial. And so one of the things that they want to do is see not only if they can continue to reduce the formation of these toxic peptides, but they also want to look at neurofilament. So neurofilament is basically a protein that's released when your neurons die. And so they basically want to see a change in the abundance of these neurofilament light chains, which will suggest that the neurons are dying less or, not dying at all. And so they're extending this study and continuing it. ASO clinical trials using antisense oligonucleotides have worked quite well for some diseases. In fact, spinal muscular atrophy, which is a disease of children, the first medicine that was able to give them hope was Spinraza, which was an oligonucleotide medicine that bound to an RNA and affected its processing. And so that molecule is approved and given to children every day. And so the hope is that the learning from that medicine can be applied to this medicine and others that are coming through the pipeline to ultimately make this not be an experimental drug, but an approved drug. but there's much work to do, as you all are well aware. And that work to do is part of because drug discovery often takes many years. And so when you start with a target, you often can test between 5,000 and 10,000 compounds to get a preclinical molecule that works, say, in a human cell or a mouse model of that disease. And you want to whittle down from these, 10,000 compounds, maybe 5 compounds that get put into a phase 1, a phase 2, and a phase 3 clinical trial to hopefully get one FDA approved drug. And so this is long, so it can last from 10 years to 15 years. But the other issue is it's risky in that you don't want to put all your eggs in one basket for one medicine, because there's going to be issues where the compounds get toxic. I mean, these preclinical studies are done in a mouse, but clinical studies are done in a human, and humans obviously are very different than mice, and you can't predict what might be. You can't predict with 100% accuracy what's going to work in a human based on mouse studies. And the Hippocratic Oath is that, we really want to make sure that whatever we do is we don't do any harm to the patients. And so what me and my group do, just to come back quickly, and I have maybe 5 or 6 slides on this, and then I'll end, but then happy to answer questions, is we don't target RNA sequence. We try to target this repeat structure with small molecules. But we want to do that in a manner where the output is just like in these oligonucleotides, you eliminate this gene. And I'll walk you through 2 cases where we do that. So just to remind you, the wave molecule worked. Here's the outline of this wave molecule. It worked by recognizing C9orf toxic messenger RNA with a synthetic piece of DNA. That synthetic piece of DNA recruited RNase H, which is basically scissors, to cut and destroy the toxic RNA. Now, when you use these oligonucleotides, they don't target the RNA structure that's causing the disease, they target outside of that. And so what we had the idea, which we've worked on for probably the past 20 years, is could we have a molecule that binds to an RNA structure that is not the part of the gene that harbors the toxin, but the actual toxic part of the gene, and then, just like antisense oligonucleotides do, recruit a ribonuclease or scissors to go and destroy that RNA target. And this is work by Jessica Bush, who I have a picture of the students at the end, and I'll call her out. She's really, I think that's one of the joys here is working with these students that are really keen to learn things, and then seeing them develop is really breathtaking in many cases. And certainly Jessica's given me that a few times. And so Jessica was able to design this molecule that has 2 ends. One end binds to the RNA, and the other end binds to an editor or an enzyme that acts as scissors to cut our RNA target. And so her and her colleagues designed that. This molecule can cut off and eliminate about 60% of the gene in human patient neurons that are derived from skin. And so one of the things that we have as technology now is we can take skin biopsies. And this was work in collaboration with Jeff Rothstein and Len at Johns Hopkins University and Len Petrucelli at Mayo Jacksonville, where we had a repository where we could take skin of patients that had C9 ALS, and you can differentiate them into neurons. And so we're able to test these drugs in patient-derived neurons. And then we could take these molecules, and they're big, so we had to inject them directly into the CNS. So these are the toxic RNA, this degrades a toxic RNA via injection into the mouse CNS. And that direct injection can eliminate, again, 60% of the gene target in the mouse. And so this is another approach, a very different chemical structure that can seek out and destroy C9orf. Now, the problem with that, well, let's put it this way. We would like to further optimize that compound. One of the issues is you have to inject it in the spinal column, and ultimately we'd like to be able to take molecules orally bioavailable, that is, you swallow them like I was, drinking my drink, or you inject them in your arm just like somebody that has diabetes would take insulin. And so in order to do that, Jessica and I had to go look at the way that C9orf RNA was assembled. And so RNAs are transcribed or made as pre-messenger RNAs that have to be assembled and put together to form what you want. And so an analogy for this is putting together a toy is like assembling a messenger RNA and a protein. So you get these parts, and just like a transformer, parts set up in a certain way can give you a solar helicopter if you use instruction 1, and parts can be set up to be a solar car if you use instruction 2. And so that's no different than C9orf RNA. And so C9orf RNA is made as a precursor where these large shapes are segments of that pre-messenger RNA that get made into protein. And so what happens is this pre-messenger RNA gets processed just like you remove the framing in these instructions and you throw the framing out. These introns, or these lines here, they get removed to stitch together the exons, and they get thrown in the trash can. But what's interesting is the G4C2 repeat is also intronic, and in a normal way, it's removed like framing and thrown in the trash can, but it's not. It's stuck as part of the intact messenger RNA. So the question Jessica and I had, is could we develop a way where we could chemically excise that toxic intron? That toxic intron could go to the trash can, and the regular messenger RNA could get properly stuck back together with a drug that we could inject in, say, your arm. And that would eliminate this toxic RNA. And so Jessica was able to do that. So she was able to make a drug, and this is a published paper. This is why Gene talked to me. So you can eliminate this intron in, again, these neurons derived from skin, whereas a C9orf pre-mRNA part that doesn't have the introns is not affected. And we do it in a manner where we eliminate just the toxic part of the protein, but we leave just the toxic part of the RNA, but we leave the protein intact, where we literally act as a surgical scalpel to just cut out the toxic part and then glue the normal parts back together. And so when we did this, Jessica was able to inject these compounds IP. So basically, you can inject these molecules intramuscularly or intraperitoneally in a mouse. And these compounds were very low molecular weight, so they'll get into the brain. And so when we inject them peripherally, they get into the brain and they eliminate this intronic RNA, and they reduce the formation of these toxic dipeptide repeats in the mouse. And so what the advantage of that is it allows us to give molecules where we don't have to directly inject in the spinal column. And it should be easier on the patients, but more broadly, it allows us a different way to go after the disease, knowing that many of these medicines that are even put into clinical trials are ultimately not going to be approved. And so, I want to come back here as I wrap up to tell you is that I think there's a lot of hope for ALS medicines. Now, we're not there yet, and we should all be very impatient, right? Because you just don't. There's one thing in life you can get back. It's not time. You can't get back time. So we all have to try to do the best we can to make a medicine that helps people and do it in a fast way. And so, there's a lot of clinical trials, and there's a lot of clinical trials that are ongoing because of genetics. We've identified many mutated genes that cause ALS. And so there are novel compound drug targets. There's novel molecules that can affect these drug targets that are being put into mice and ultimately humans. In addition, as I touched on earlier with regards to this nuclear pore, biologists, by studying this disease, because the gene, they can make model systems to figure out ways in which the biology is affected, where you can make medicine into affect an unprecedented pathway, such as the nuclear pore. And I think it's hard. All clinical trials are an experiment. There's going to be failures along the way, but by collecting the data for the experiments, you make yourself smarter and you make yourself stronger to do the next clinical trial and ultimately get a medicine to be approved. And I think ALS is so critically important here, not only for paving a path to develop precision medicines for ALS to help patients that have the disease, obviously. But we're also thinking about other neurodegenerative diseases. Alzheimer's. Alzheimer's is a neurodegenerative disease, but it's not as clear for what the targets are in Alzheimer's, I think, as it is in some of these ALS diseases. The other thing is, as more compounds are being put in ALS trials, it'll pave the way for better medicines for the whole field of developing medicines for repeat expansions. And vice versa. As I mentioned earlier, and I was asked earlier, there's multiple medicines for Huntington's disease in clinical trials. There's multiple medicines for type 1 myotonic dystrophy. Most certainly, and when readouts of the data for those approaches come, there's more experimental data, it'll accelerate our knowledge, the depth of our knowledge base to more rapidly develop ALS-associated medicines. And so I think all of this is good that it's coming. And, scientists and physician scientists need to be diligent to use the data in a smart way to make sure that we're faithful to try to get a medicine that's as effective as possible. And it's that medicine is developed and discovered as efficiently as possible. And so most of the work today from my lab was by Jessica Bush. And, I told you earlier, my output here in lab is the students of the science. And I want to tell you that Jessica's a really amazing person at many levels in terms of her ability and capacity to do amazing science. But I think Jessica, in this regard, illustrates for me how lucky I am to be able to work and learn from these people on a daily basis. And also, you guys. Thanks, Gene, for giving me an opportunity to talk to you folks. I'm happy to answer any questions you have. And, I certainly use these talks as inspiration to do the best science that we can together. So if you guys have any questions, I'm happy to answer them. And I also want to thank the US taxpayer again. I mean, that's really critically important. Scientists complain they don't have enough money, but there's resources to do very challenging things. Find a gene in 2011 that causes ALS and FTD, which is unprecedented. And I think one of the other things that I'm lucky about in doing science is every day you learn something new. I think as a scientist, sometimes we take that for granted. But every day in the lab, if we do it right, we should be learning something that no one else has known before. And when you're doing that, where it's understanding the impact of a disease on a human, it's pretty profound. So anyway, thank you very much for taking the time listening to me. I'm happy to answer any questions you guys have.
Wonderful. Thank you so much, Dr. Disney. We're all so glad to have heard this. We did have one person already with a hand up. Jim, do you want to go?
Yeah, thank you. And this is so inspirational. Really, really appreciate both and the legacy and the work that your lab is doing, Dr. Disney. This is just great. I have a question. I'm in the Wave 2A 1B trial, and I'm curious, based on what you've seen of the methodology and what they've said, about what's the sort of best one could hope from that sort of approach in terms of both effectiveness given the low dosage and the potential toxicity of taking a lot of it. Thank you.
So I have no idea how to answer that question. I know that the dosing that was given in the WAVE trial, can I just stop presenting? Because then it's easier for me to see everybody. Thanks for the comment. It's an experiment that WAVE is going to have to conduct. I'm grateful that you're in that trial because I just don't know how to answer your question. That's why they have to do the experiment, Jim. And what was promising for what I saw this morning when I reminded myself of the data is as the dose went high from 10 mgs to 30 mgs, the drop in the toxic dipeptide repeat decreased even more. And so what they're going to have to gate in this trial, and what I could guess from the very small press release that I got, is they have to figure out at what dosing they can get the maximum knockdown without having overt toxicity. And that's an experiment that that has to be run to figure it out. But I think there's very profound lessons to be learned here, and I'm, very grateful for you to be participating in it.
Oh, absolutely. By the way, they've set for this phase the dosage at 20 milligrams, not 30. And unclear at this point if it's making a difference, but I'm trying to get my own DPR, NFL results to try to understand whether it seems to be working. So thank you. Appreciate it.
No, thanks, Jim. Take care of yourself.
Just want to say, the doctor said thanks, Jim, and all of us who are impacted by C9 appreciate everyone who is in clinical trials of all kinds. So we're thankful as well. Jeremy had his hand raised.
Thanks, Gene. And thank you, Dr. Disney, and for all the work you and your students are doing. A lot of your presentation went right over my head.
I'm sorry.
No, that's fine. It's me, not you. But this question may be a little off base, but some of your explanation of the cutting and putting back together RNA sounds to my layman mind very similar to CRISPR. And I'm kind of curious if you can help distinguish the two in my mind.
Yeah, great question. Thanks. Yeah, in some sense, so CRISPR is as you guys know, CRISPR is a gene editing tool where it's basically a large protein nucleic acid complex that basically seeks out genes, and it can replace genes and edit them. They're tremendously big molecules. And so that if they're tremendously big, it's very hard for those compounds to get the CRISPR to get a large tissue penetrance to really erase a lot of the gene. But they're working on it, and I'm optimistic they'll be able to do it. Our molecules are able to, they're very small compounds. They're order, hundreds fold smaller. And because they're small, they're able to get in higher amounts into affected cells and tissue and have a broader spread. And so even though you can imagine that they have a similar functional output, which I agree with you, there is some similarities in their functional output. The small molecules, at least for this indication and at this stage, can get into more tissues, and in principle, although we're not there yet, could be more efficacious. But you have a long way to go for that. And even the oligonucleotide approach of WAVE is much smaller than a CRISPR approach, right? That's going to get into, in principle, more tissues when it's delivered centrally than CRISPR, that has a lot of extra cargo that's associated with it to even get it into certain cells. But it's great that we're even talking about this, because I couldn't have imagined 10 years ago having conversation with people and them knowing state-of-the-art techniques that I use in my lab, like CRISPR. And so I'm just amazed at that. So thank you, Jeremy, for your question.
Thank you.
There's one other hand raised. Before I it to that person, I did just want to ask, as a privilege of, I guess, of the host a little bit here. In the ALS M&D symposium, Stephanie Frechette from Biogen shared that perhaps they sort of looked at the preclinical data from their two ASOs, tofersen and the C9 one, and they said that the C9 one, of course, had the poly-GP lowering in the mouse model, but did not have NFL lowering, where tofersen had NFL lowering in the mouse model. And I was wondering if you have anything to share about that. That might be too in the weeds.
No,
but
so the other ASO is the SOD, superoxide dismutase tofersen, right? Okay. I don't know specifically. I think it's hard for me to be able to answer that with rigor, because we're just getting a press release from the C9 ASO. Now, what has been suggested for me, which goes back to one of my earlier slides, is that there's both a sense and an antisense transcript. And so some of the feedback that I've heard from luminary biologists in this is that you need to have a compound that can silence both the sense and the antisense. But I would like to, one of the things that I think is so critically important is that when the top line data is released, I think very quickly Biogen should be giving detailed data in the publication so that scientists can digest that information and come to their own conclusions. Because when that top line data was released, I wanted to dig in to understand what exactly was going on. But if they're not releasing data, it's very hard to do. And I know that they'll ultimately do it. It's just that we'd like to see it sooner. And these are different diseases. I mean,
totally.
Right. I mean, it's funny, you hear cancer and you think it's one disease. You hear ALS and it's one disease. It's not. They're very different diseases, and it just could be a function of the different ways the disease manifests. That one NFL is down and one isn't. I don't know enough to be able to comment on that with rigor.
Yeah, no worries. Also, if there's any specific questions you have, we do talk to them, and we can try to push them on it as well from our angle, because of course we want to have those questions answered as well. We did also have a question about where you're looking at this small molecule in relation to development. Are you partnering with pharma yet, or doing anything like that to get into humans? Or
yeah, I have a biotech company, Expansion Therapeutics, that's looking at these and other molecules for C9 ALS that are binders that are going after it. So yeah, there's a 30-40 team group that's taken these discoveries and trying to blow them out. And they're trying to do it in a way where the premise is to do no harm. So they need to go through a lot of derivatives and make compounds to see which ones are the safest. And then hopefully they can get into clinical trials. So, yep, that's what we're doing.
Wonderful. That's great. I see the other raised hand, and then Amy or Mindy after this next question, if you guys want to ask your question too, or if you want me to ask it, whatever you guys think. But the Griff family,
yes.
If you want to go ahead.
I'm just wondering if you have any idea when you might go to a phase 1 trial in humans.
Oh, I have no idea.
Yeah.
We would have to get a compound that had compelling phase 1 data to be able to do that. Now, we want to compress the timelines and do it as quickly as we can, but we don't want to do it and then have it fail, right? So that's the balance.
Yeah.
So I don't know. I'm not good at predicting stuff like this. So
okay, all right. But you are moving towards that, I imagine.
Oh, that's the dream, right? So we have to try hard to make that dream be a reality, right? Yeah.
Okay, great.
We had two people in the chat, Amy and Mindy, both ask about symptomatic treatment, and you were looking at your slides, but I mentioned in models often they're treated pre-symptomatically, is my understanding. Have you given a consideration to, we're so far away from you in trials, but in the event, do you think that this should be looked at both pre and post symptomatically in C9 people?
Wasn't that the reason that the SOD compound was able to get more efficacy, is that they gave earlier patients the drug, right? And so in principle, yes. The question becomes, are the biomarkers for reducing DPR going to be sufficient to do the clinical trial on it? And how early do you have to go to prove the efficacy? Now, it's ALS, and so the FDA is very likely to not slow down drug development to have all the answers. But yeah, I think ultimately you want to take these things pre-symptomatically. And, you know, Biogen, I think there's data with SOD to show that the earlier you go, the better the effect that the drugs have. So, but we'll have to see what happens. I mean, the FDA, they're slow, but I do think they try to make the best decisions with the data that they have at hand, right? And I know it gets frustrating, but I do think that they try to do what's right with what information they have.
Okay, everyone. Well, I guess that's it then. Very happy to have had you, Dr. Disney. Again, we are Genetic ALS and FTD and the Legacy. I'll put our little new website link here again so people can see that. We are trying to make a difference for our community, at the very least, by making sure we have information. So have a good night, everyone.